Alright, and now on to the major apolipoproteins. The apolipoproteins are amphipathic proteins, which just means that they have both a hydrophilic as well as a hydrophobic portion that bind to lipids to form lipoproteins. Now this is done so that lipids can be carried through the circulatory as well as lymphatic systems. Now there are 5 important and most tested apolipoproteins, and we're going to cover each of those here. First off, we have apoprotein E, which mediates remnant uptake into the liver. Now ApoE can be found on VLDL, IDL, HDL, and chylomicrons, as well as chylomicron remnants. Now what you need to remember here is that ApoE mediates the uptake of these particles at the liver. And also remember that ApoE is found on every lipoprotein except LDL. Now next up, we have ApoA1, which activates LCAT, and this is found on HDL. Now LCAT converts cholesterol into cholesterol esters in the HDL molecule, so it makes sense that ApoA1 is found on the HDL lipoprotein. Now next up, we have apolipoprotein C2, and it's the cofactor required for lipoprotein lipase. Now remember, lipoprotein lipase is used for degrading triglycerides and VLDL as well as chylomicrons. And as we'll see later on, chylomicrons and VLDL are loaded with triglycerides. And since they have ApoC2, they can find lipoprotein lipase on vascular endothelium and unload some of their triglycerides into surrounding muscle as well as adipocytes. Now you can also find ApoC2 on HDL. Next up, we have apolipoprotein B48. Now this mediates chylomicron secretion, so it's only found on chylomicrons and their remnants. Another important point here is that it's only found on lipid particles that are originating from the intestines. A good way to remember this is that the last part of ApoB48 sounds like the word 8. Now our last major apolipoprotein is B100, and it's mostly present on VLDL, LDL, as well as IDL. Now ApoB100 binds the LDL receptor and allows for these lipoproteins to unload their products into cells within the body. Also remember that ApoB100 is only on lipid particles that are originating from the liver. Now one more fun fact before we go. ApoB48 as well as ApoB100 are made from the same gene. So then how are they actually different proteins? Well, this is an example of alternative splicing, and the mRNA for these lipoproteins is processed differently, depending on whether it's from the hepatocyte or the enterocyte. Well, alright, that'll wrap up major apolipoproteins. See you in the next video.